Welcome everyone. Thanks for coming along to this next ACTA Adaptive Platform Trials virtual clinic. For those who've been at these before, it's an opportunity for us to hear about a new platform trial, adaptive platform trial, and for panelists' feedback and also audience participation. My name's Steve Tong. I'm an infectious diseases doctor and a clinical trialist. I'll quickly introduce the rest of the panel and then we'll acknowledge country and ask Aidan to get started. Really privileged today to have a great panel. So there's Arlen Wilcox, who's from the NHMRC Clinical Trial Centre at the University of Sydney. He's a clinical trials program manager and leads the expansion of novel and adaptive trials methodology. Dr. Robert Marr is a biostatistician at the University of Melbourne, has a focus on Bayesian methods and novel experimental design and analysis, particularly on adaptive platform trials. There's Dr. Hannah Carter, who's a health economist and senior research fellow based at Queensland University of Technology at the Australian Centre for Health Services Innovation. And then Mitch Messer, who's a consumer representative and part of the community engagement team at the Kids Research Institute in Perth. And really importantly, providing us with a consumer voice today on the panel. I'd like to acknowledge the meeting today. So we'd like to begin by acknowledging the traditional custodians of the lands on which we meet and to pay respects to the elders past, present and emerging. And we'd like to extend that respect to Aboriginal and Torres Strait Islander peoples who are here today with us on this webinar. Without any further ado, a little bit of housekeeping. Please make sure that you're on mute. Put your hand up if you'd like to speak during the Q&A session. And there's also a Q&A box if you want to type in any questions as well. The session will be recorded and it will be made available on the ACTA website. So without any further ado, we'll hand over to Aidan Burrell. Thanks, Aidan. And maybe if you'd like to, no, I haven't got your CV up here. Maybe if you'd like to just quickly introduce yourself, Aidan.
Okay. Well, thank you, Stephen, and thanks everyone for a fantastic opportunity. I haven't been on one of these before, and hearing about that introduction, I'd like to join more of these. I will just check that firstly, the slides are coming up okay, and you can hear me all right?
Yep, all good.
Okay. So, yeah, look, I'd like to thank everyone for this opportunity. My daytime job is I'm an intensive care doctor in Melbourne, and I also work at the Australian New Zealand Research Centre at Monash University, which is a critical care methods centre looking at providing clinical trials and methodological support. And the trial I'm going to talk about today is a trial that's very dear to our hearts as intensive care doctors. We're looking at respiratory failure, and we're looking at particularly a subgroup of respiratory failure called ARDS, and I'll go into that a little bit in a second. This is a collaboration with an international group in the UK who are leading this, and we're interested in bringing this platform trial to Australia and as part of a bigger collaboration with a lot of work that we do together, looking at trying to bring the sort of the power and the advantages of platform trials into this particularly tricky area to research. So just to start off with some disclosures, we are working with a company that's going to provide the point of care testing that I'll go into in a second. So there is some IP and there is some commercial support, and it's been provided by the company for free for the trial. Otherwise, no other disclosures. Okay, so acute hypoxic respiratory failure is the broad category of patients who are hypoxic, who have evidence of lung injury, and it's an acute syndrome, so it's sort of less than a week in onset. That's the broad category, and it's a common category for any clinicians on the call will recognize it, and particularly in the intensive care, this is a very common syndrome. There's a subset of these patients who have a syndrome called ARDS, which is an acute respiratory distress syndrome, And this is a heterogeneous syndrome where the lung capillaries become injured and they become very inflamed, and fluid leaks out of the lungs and into the alveoli. And it leads to a lot of inflammatory injury. It leads to a lot of hypoxic respiratory failure. So when I'm talking about ARDS, I'm talking about a subgroup of patients. It's usually between about 10 and 20% of the total patients with hypoxic respiratory failure. As you can see, this particular subgroup has a very high mortality. Once you develop this syndrome, then you're looking at a mortality rate of about 30 to 50% in the largest observational study done several years ago now, looking at this group. And, you know, the treatment usually involves mechanical ventilation, a lot of intensive treatments, and this leads to a lot of prolonged ICU care. Survivors have a lot of long-term disability and physical and cognitive impairment. And it's incredibly expensive syndrome. In addition to the mortality and the morbidity, it's a very expensive syndrome because patients tend to stay in the hospital for a very long time, not to mention the ongoing sort of disability and inability to return to work. So that's just sort of a bit of an introduction about the disease and the problem. And really, since the beginning of ICU, people have recognized this, and they've recognized that, you know, really we need pharmacological treatments to try and, you know, stabilize or even reverse the process. And you can see there many things have been tried. And the problem is, while they have really great kind of biological or theoretical mechanisms, unfortunately, there are no proven treatments for ARDS, and nothing has really worked so far. And part of the sort of problem is it's a very heterogeneous disease. So in ICU, we often deal with syndromes, and by that I mean a constellation of symptoms that we kind of put all together, and we call it ARDS, or we call it hypoxic respiratory failure. But you can understand that the disease underlying what the lung is causing the disease can be very different when you've aspirated, or when you have a very severe COVID infection, or when you have a severe septic shock and your lungs get weak and leaky that way. So although we bunch them all together, the underlying mechanisms that cause it are very different, and these have very different impacts on which drugs to use and which, you know, what sort of approach we have to treat it. And the idea, a central idea for this trial, is to use the idea of precision medicine to reduce heterogeneity in this syndrome. And maybe it's actually by reducing this heterogeneity that we might find that drugs work in certain types of ARDS, but not in others. You know, in some other types of ARDS, the drugs may be harmful. And it may explain why we have drugs that in general don't have any effect, but within subgroups can have benefits or harm, but an overall neutral response. And you can see here that, you know, the old paradigm of syndromes of respiratory failure, ARDS, sepsis, actually moving forward, we identify using biological mechanisms about different treatable traits, which we can then go on and test these drugs. This, we think, is probably a key insight into how we're going to tackle these diseases moving forward. And so I mentioned precision medicine. What does that exactly mean? So by that, we discover clinical phenotypes or biological phenotypes. And then what we do is we develop point of care tests. So in this example, it would be blood tests, which we can then look at these phenotypes and prospectively validate them by using different drugs that already exist, by repurposing them or developing new therapies and testing them in these subphenotypes rather than testing in the whole overall syndromic group. And so this is really what individualized critical care medicine is. We've, you know, in hematology and certain cancer diseases, and, you know, this has been around for longer, and I think it's a really important step for critical care research follow suit. Latent class analysis is a statistical and analytical method of looking at trying to understand and identify these subgroups from within broader syndromes. You can see here that it's been around for a while, but it's really only been in the last decades that the statistical analysis and the sort of computing power has been advanced enough that we can actually apply this to bigger populations. And so one of the real leaders in this has been a group in America. Carolyn Calfee has looked at the ARDS population and has found in repeated independent populations really two main subphenotypes, which have been shown, as I said, in independent groups that seem to have a differential response to both different biological mechanisms and also differential responses to treatment. Phenotype 1 is called the hyperinflammatory ARDS syndrome, and they tend to have lower bicarbonate, protein C, and systolic blood pressures. And then phenotype 2 is the hyperinflammatory group, making up about a third. And this group tends to have higher interleukins, tumor necrosis factor, and various other biological markers as well. So this biological signature, in fact, appears to, we seem to have these two separate groups that are ripe for further investigation. And when we look at these groups, the hyperinflammatory group, you can see there that these groups have quite large impacts on outcomes. So day 90 outcome in the hyperinflammatory group of ARDS is around 19%. The hyperinflammatory group has a mortality rate of 51%. You can see that ventilator-free days, organ failure-free days, these are markers of the amount of support that patients have in ICU, are vastly different. So the hyperinflammatory ARDS does definitely have prognostic information. But is that all? So what's been developed over the last couple of years from the Calfee group has not only does it have a prognostic importance, it also, these subphenotypes respond differently to different drugs. And one of the best large overall studies is this study looking at the response of the hyper and hyperinflammatory phenotypes to simvastatin, which is a common anti-cholesterol drug that has other anti-inflammatory pleiotropic effects. And what you can see here is that in the hyperinflammatory phenotype, simvastatin appears to reduce the risk of death, but in the hypoinflammatory, it has no benefit at all. And so you can see if you combine all this together, you often get neutral results, but it's by subphenotyping that we see the benefits, potential benefits. And this analysis has been repeated looking at other interventions, including steroids, including fluid resuscitation, including and being repeated in multiple studies. So it does seem like there is this, potential differential response that appears to be reproducible. Another barrier in the past has been, in order to do this, you need to take a blood sample that is rapidly able to be analysed so the clinicians at the bedside can actually apply these phenotypes. And really over the last sort of 5 to 10 years, the point of care devices have exploded in their capacity to do this sort of research. And so we can now look at IL-6 and TNF alpha and bicarbonate, and we can actually define these subphenotypes right as they come into the emergency department. And we can actually randomize patients based on this right from the go. Okay, so there was a bit of background there. Apologies if I went on for a bit long, but I wanted to sort of set the scene for why this trial is a bit different. So I'm now going to talk about the Panther trial, and this is a international phase 2 open label precision medicine trial that uses Bayesian Adaptive multi-arm trial design, and it's been built over the last sort of 3 years incorporating all the background I've just explained, and it's developed some novel methodology around the identification and randomization of these patients based on their initial phenotype. There are a few design features that I'll be going through. The interventions that we have decided to look at have been created through an independent intervention prioritization committee, which liaises with the main study management committee. There's been a lot of early career research engagement. I'm an early career researcher, and there's been a whole lot of work done in that space. There's the commercial collaboration I mentioned with the device, and there's going to be a whole range of other embedded biobanking and consumer involvement as well. So the aim of this Panther trial is to establish an international phase 2 precision medicine adaptive platform trial to test the efficacy of prioritized pharmacological interventions in patients with different phenotypes of acute hypoxic respiratory failure and ARDS. The secondary aim is really to, once treatments have been identified to work, then we would use this to feed into other trials that can then do the phase 3 testing. So this is a phase 2 platform trial. The research question is looking at adult patients with acute hypoxic respiratory failure with either a hyper or hyperinflammatory phenotype. The two treatments that we're going to start with are simvastatin and baricitinib. The control is going to include standard care, and our primary outcome is going to be day 28 organ support free days. I'll go over these in a bit more detail over the next few slides. So we would like to include, in patients with ARDS, we'd like to include all patients with ARDS. And there is a definition that I haven't actually included here, but it involves bilateral infiltrates on the chest X-ray, a degree of hypoxia of what's called a PF ratio of less than 300. We measure the degree of hypoxia, and it also has to be acute, so we can't include patients with chronic bilateral infiltrates because that's not an ARDS picture. We also have some exclusions. Patients less than 18, greater than 48 hours from the acute hypoxic respiratory failure diagnosis, patients who otherwise would be withdrawn, previous trials, domiciliary mechanical ventilation, or declined consent. The additional intervention-specific exclusions, will be included as well, depending on the particular intervention. So I mentioned this before, we have this independent prioritization committee, and you can see the way that these are selected. So we have the trial team, the academic partner, and an industry partner. Different interventions are proposed. They go to the committee, and there is this internal process that's independent of the trial to come up with the the highest priority treatments. And if the committee rejects them, then they're rejected, and then eventually they're presented to the trial steering committee. So this is all published online. You can see the address there, and you can see on the right-hand side at the moment, these are the treatments that we're going to look at: simvastatin and baricitinib, and then we have 4 others below, various immune modulation treatments that we would look at following the initiation of the first 2. I'll go over this just quickly, just to say, as I said, simvastatin is a HMG-CoA reductase inhibitor. It's commonly used to treat hypercholesterolemia. There is some preclinical data and phase 2 data to support its use, and the analysis I mentioned earlier about there being a signal towards benefit in the hyperinflammatory subphenotype, but that was observational data only. In REMAP-CAP, we looked at patients with COVID and simvastatin, and we found that there was a 95.9% probability of superiority compared to control. Didn't quite meet our predefined sort of limits for superiority, but it was very, very close. And in general, we found it to be well tolerated even in critically unwell patients. Baricitinib is an oral JAK2 inhibitor, and it's commonly used in autoimmune diseases. It inhibits a range of different inflammatory cytokines, all of which are involved in the pathogenesis of ARDS. So we think it's a good targeted anti-inflammatory drug that will hopefully provide benefit, and it's generally been well tolerated in other trials. So these are the first two that have been prioritized. The approach to the trial is to perform regular adaptive analyses, and then over time we'll identify those treatments that provide benefit across the different subphenotypes, and then we'll equally look at particular interventions that are not responding or meeting our futility criteria, and then they'll be stopped to make way for other interventions. The allocation will be concealed, randomization will be in a 1:1:1 ratio, and stratification will be done by phenotype and also by site. We plan to co-enroll with other acute respiratory failure and ARDS trials. The space is pretty busy, but we've done a lot of sort of background work with some of the existing work already in this space, and the idea is that, in fact, Panther will actually become an arm of REMAP-CAP, which is another major platform trial, and it will work with other trials as well in a very collaborative and even federated manner in order to maximize really every patient is being enrolled in a trial and is getting the maximum benefit out of that. Our primary outcome is organ support-free days to day 28, and this is a commonly used outcome used in critical care trials. It's a scale, so on the scale you can, the worst case scenario is if you die in hospital, you get a -1, and then from that you then look at the number of days where you're not requiring organ support, and you subtract the number of days you're on organ support, you subtract that from 28, and then the larger the number, the better you've gone. So the ideal would be no days of organ support, and so therefore your score would be 28. We've done some work looking at this primary outcome in this ordinal scale, and it correlates very well to long-term sort of outcomes, day 180 outcomes, and is also, we've done some work with our consumers who have supported this and have been involved in the development of this as well. So I think this is a good primary outcome for this sort of work. There's a whole bunch of secondary outcomes. I won't go through them all. These are pretty standard for critical care trials. I guess I'll just draw attention to health-related quality of life at 90, and also serious adverse reactions will be important. This is a busy slide. I don't know if it's projecting very well, but it just shows how, if you look on the left-hand side, this is a patient coming in. This is the sort of flow of what will happen. So they will get their point of care testing right in the emergency department or early on in the ICU. And they, based on that point of care testing, they'll either be moved into the hyperinflammatory group or into the hypoinflammatory group. And then from within these subphenotypes, they'll receive either usual care, simvastatin or baricitinib, and the same here. We will continue to enroll patients, perform adaptive analyses, and then we stop when the intervention either is indicated as being efficacious or futile, or when the arm reaches the maximum sample size. This can continue on with our adaptive analyses until we reach this, and you can see how it goes. And as I mentioned, if futility is reached, the arm is dropped, and then we can look at including other potential interventions as well. The statistical triggers we have, I'll just draw your attention to this, So if you have an odds ratio of 1.05, so showing more than 5% benefit, and if you have a greater than 88% chance of finding this, then that's been defined as being efficacious. If you have a greater than 70% chance of having a less than an odds ratio of 1.1 of benefit, then we're calling that futile. And for people who are used to looking at these, you'll notice that the triggers, the percentage of certainty that we're using here are quite a lot lower. And this is to reflect the fact that we're looking at a phase 2 trial, and the idea is we're not looking for hard and clinical points. We don't need as much certainty as a phase 2. The idea is once they meet those triggers, we will then feed them into larger trials that can then look at those clinical outcomes. I'm happy to talk afterwards about this if as well. Okay, we've done a whole bunch of simulations and sample size calculations. We think based on our sort of existing data that we expect about 952 patients will be enough for the 3 different interventions. So the control arm, the simvastatin and baricitinib, as the trial begins. If you look at the maximum, we've got a maximum of 956, and we've capped it at 2,163. Looking at 70 sites, if we are recruiting between 1 to 2 patients per month in each site, we think that this is achievable in 2 years. We have up to 4 years to recruit that, and we think we're hopeful that we've come up with a conservative approach that should lead to results reasonably quickly, hopefully a lot quicker. Just I think the last 1 or 2 slides. The group in the UK have a very advanced consumer group, and consumers have been involved all the way. In Australia, we are developing, we haven't actually developed as far a lot of this work. So what I'll mention is work that's already been done in the UK. The James Lind Alliance Priority Setting Partnership in the ICU, one of the key things found out from this priority setting was what is the best way of preventing lung damage in patients receiving respiratory support. And we think that this trial directly addresses that well, and it's a high priority for consumers. And then we've also, as I mentioned, the group in the UK has done a lot of work already with the consumers, looking at the potential benefits, particularly in concerns around long-term improvement in long-term disability that occurs following ARDS, and the great potential here. But there's more to be done in this space, I think, before it completely comes to Australia. In Australia, we're looking at starting with 10 sites to begin with. I just mentioned the consumer engagement process that we're just doing now. The point of care device will be supplied free of charge by the company, and we are anticipating a waiver of consent for the initial blood collection, and then a prospective consent model within 48 hours. We were aiming to submit for a grant just in the last month, but we're going to aim for early 2025 for grant submission. And so just my last slide. So Panther trial is a highly novel precision medicine phase 2 adaptive platform trial, and this fills an important gap in the ICU and even sort of broader community. The biological rationale for subphenotypes within this group is strong. There's some strong observational data suggesting this, but we don't have any randomized data to support this, and that's what this next step will be. The prioritization group is starting with simvastatin and baricitinib, both drugs that are well known to intensivists and we think are feasible. Consumer engagement, I've mentioned. This group is a very experienced trial group, and we hope to really have a pipeline for novel drug testing into the future, phase 3 trials. So that's the end of my presentation.
Thanks, Aidan. Fantastic. Really good to hear about this reasonably novel approach, and particularly bringing in the precision medicine and kind of devices in as well. I might just start with a couple of quick questions. Is that point of care device, is that an already approved device that's being used in kind of clinical care at the moment, or what's the kind of pathway for like, obviously it's quite an integral part of your trial. Do you know if the company which produces it, are they heading towards kind of registration for this kind of use? How do you fit in those kind of industry related priorities for them?
So it's already in use for the use of toxin testing in Australia. In the UK, it's registered for this use. I've been engaging with the Australian arm of the company, and I have to say I'm not an expert in a lot of the regulatory stuff, but I've been told that it shouldn't be a barrier. It's already in use, clinical use. It's just we're going to look at a slightly different application, looking at inflammatory biomarkers rather than toxicology. But yes, that is it's a good question, and I think it needs to be completely sorted. But I've been told it should be fine.
And I'm kind of interested in, you could probably choose a couple of points at which to do the randomization. You could randomize to either getting the point of care device or not, and then following an algorithm, as opposed to where you're saying everyone gets the point of care device and then there's randomization for different agents. Was there any consideration of that kind of earlier, almost in the algorithm itself includes or doesn't include the use of such a device?
I think that's a good question. I think our strongest rationale and belief is that there is going to be a difference between these phenotypes. I guess it will almost be like a control arm. In addition, a control arm with the device, and then the device with the phenotypes. I think it's a good suggestion. I mean, my concern is we have a lot of data already in the control, in a kind of a control, and it would require some thinking about, well, how would we manage the treatments in those patients? The treatments have been tried in a lot of non phenotyped patients and found not to have benefit. But I think it's an interesting thought, because that could provide. I guess it would be hard to make the argument that we are going to benefit them if they've already been tried in these trials and not had an effect. But it's an interesting thought. I'll give that some more thought. I think just from a clinical point of view, we think it will be easy to apply. It's just a small blood sample. It's very easy to actually do. So we're hoping that once the trial's up and running, it shouldn't be a barrier in itself. Maybe for example, if there were patients who are eligible who for whatever reason are not included, we should collect data on those patients as a kind of as a registry and look at the outcomes of that group as well. Maybe that's another idea.
Maybe Mitch, do you mind, from a consumer perspective, what are your thoughts at the moment?
Yeah, no, I'm good to see all that work that's been done in the UK, because I know the UK are very good at that sort of make sure that there's people involved in that, and the James Lind Alliance and where that's come from. You mentioned that you're starting to do that. What is your plan in Australia? Because you mentioned you're going to do something, you're going to have consumer engagement, but tell me how you plan to go about setting that up.
We've developed a consumer group of critical care, of people with lived experience and experience in critical care consumer work. And I think we, and the idea would be to look at some of the methodology here and approach that group and see if, how can we will it work? How can we improve? Is there anything we can change? I guess that's from a methodological point of view. I think, I think some people have, I think there's variation across countries and probably within countries about biological sampling. Some people are very for biological sampling, some people are against it, and we would need to, I think, explore that in our group and see how people feel about it for research purposes. So I think that's some work. I think there's, I mentioned a model of consent that might work. I think we have discussions with that group as well about the model of consent, because we haven't really landed on something yet.
Yeah, I mean, from that point of view, the lived experience too about being in that situation, and you said that model of consent and how you would go about that. Also, I guess how you'd explain what a platform trial is, or especially when you're starting to use Bayesian adaptive trials. Having been involved in a few, and whether the community don't necessarily understand these are different. So asking them about how you go about talking about how these sorts of things are different and how you can make it changing. I use the analogy I've been using recently is a trial is you set it up and it's on train tracks, and you start, you finish, you look at it. And I used to use it like you're using a compass. You'd start the trial, you have your endpoint, but then you're using a compass to guide you. But now I've sort of changed that to saying, well, people don't even know what a compass is anymore, potentially. So, it's like using your GPS and you've got 5 different routes, and you go according to that. So getting the community part about so that the information that you've got is really clear and easily understood.
I think it's a really good point. And we so in addition to the patient group, a lot of the care providers also struggle to understand some of this. And we have nursing staff, and we have other researchers in our consumer panel, but I think there's a lot of sort of lack of understanding. Oh, sorry, you go.
No, I was just clearing my throat. Go on, Gary.
The other thing I'm really excited to share. This is not published yet, but the group has just completed a survey of, I think it was 8 countries, looking at is predominantly around pandemic preparedness and acceptability of research in patients in ICU. But there was information in this survey looking at adaptive platform trials as well. I think it was about 5,000 or 6,000 people in this big survey. And I'm really, I've seen the sort of overall results. I'm really interested to look at the Australian results. There were 700 people from the community involved in this survey. And I think this is really going to give us a bit of a feel for, what is acceptable for patients and families when, for example, they come in critically unwell, they can't consent, and we go to families. We've asked them, not in that acutely stressed environment, we've asked them just, as part of a survey. And so I think this will really help help inform what will be a good design for the trial.
Yeah, I think you're right. And just, I agree there. If you can actually get this, the explanation of this for community, you'll actually get the explanation of that for all of the groups. And I have seen that where clinicians and other people doing other types of work don't actually understand what you're trying to do, and they can't get their head around it. And I've seen that from a project I've been involved in, where one of the sites was having issues with making sure that they collect data in the way that they're supposed to collect data, because it's different, in terms of their timeliness and all those other things that you need. So.
Yeah.
Mitch, can I just pick up on that? I'm just interested in thinking, how much do you really need to explain the concepts behind an adaptive platform trial as such to the patients and consumers, as opposed to saying, well, this is a clinical trial, we're trialing treatment A versus treatment B. in an area which we don't know which one works better. How important does the whole adaptive aspects? I can understand with response adaptive randomization, perhaps, where you might be tailoring more, successful treatments to more patients, but where that's not being used, how important is it?
Look, it's probably less important because that part of it's not the bit. But I think, Steve, when we're talking about making sure that we want people to participate in research, I think it doesn't hurt if we can find easy ways to explain to people the differences of what's going on, especially if they start talking to other people about it and get confused. So you might say, well, that's not the critical part of it. But I think having a general understanding, having really easy information to take on board is a good thing across the board.
Yeah, great. Maybe we'll go to Rob for any statistical comments or questions, Rob.
I'm sorry, I have to drop off because I've got another.
Yeah, thanks for your time, Mitch. We very much appreciate it.
Okay, bye.
Yeah, no, it looks like a, believe it or not, relatively straightforward platform trial, which is a good thing. I had a couple of questions just about the. I didn't see any description of the type 1 error or power of this design. Do you have? And I just wanted to sort of think about how that relates to the decision, the statistical trigger that you've stated there. So, do you know what the power is for that sample size?
Look, I will have to be honest, and I can't give you those exact numbers. It's still preliminary sort of analyses, and the sample size and the some of the modelling is I think is still being developed. That what I showed you was the initial plan. So I'll just have to get back to you on that one. Really, in reality, I do know that it's gone through a very experienced group who've done a lot of modelling, and they've got the preliminary data they're basing it on is based on other similar sort of cohorts. So including data from REMAP-CAP looking at simvastatin and also baricitinib. So we do have some already high quality data from similar cohorts of patients. But apologies, I can't go into more detail.
That's all right. I guess it's kind of related. The odds ratio being greater than 1.05, having a probability threshold of 0.88. How were they chosen? I mean, it typically will. There's two ways of doing that. One is you choose it to give you the desired power for your design. The other way is to take a much more Bayesian and clinically pragmatic approach and to set it in terms of what you want to see and how much you want to, like how confident you want to be in what you see. I just wanted to see if you could give a rundown and how you got to that statistical trigger. That's specific.
Look, again, that's a very good question, and I know a lot of work was done for those areas of certainty, and I did see some other people online who may also want to comment, but I guess I can sort of talk about more in general principles that when as a clinician we have a certain threshold for certainty which is much higher. And as a phase 2, we're accepting a lower threshold. How exactly those thresholds were defined, how much engagement in the community, I'm not 100% sure, but I know it's not been plucked out of the air. There has been some development, but I think they're both excellent questions, and I will have to get back to you on them.
Because the only thing is with the how it's set up now. The threshold looks lower, but you've got you're hoping to see an odds ratio not only greater than 1, but 1 plus something else. And so you actually might end up back at square one in terms of, if it might be equivalent roughly to the sort of standard probability of an odds ratio being greater than 1 being more than 95%. So I think it's just maybe just food for thought in that respect. And I'm sure whoever's doing the simulations has sort of their finger on the pulse there, but it's just stood out to me as I'm not sure I understand the rationale of it, and it's pretty critical to know what your decision making framework is, and to be confident that it's the right one. So just food for thought. And the other question, I guess, was, is there going to be any sort of borrowing methods used between the two subgroups? So between the hypo and hyperinflammatory subgroups, will there be any information sharing or?
There almost certainly will, but as again, a lot of this is still being worked out at this stage. I think we would have to be careful because we're talking about the ARDS syndrome, but we are talking about two subphenotypes, and we could easily, kind of muddy the waters by borrowing too much and making assumptions. But I know there's discussions around that, and I think it's in development exactly what the approach will be.
Mm-hmm. Yeah. I mean, those were the main things that jumped out. Just thought I'd give you a light grilling on. Yeah, but overall, it looks like a well considered, as I said, relatively straightforward platform trial in the context of what else is, which is as I said, a good thing, and probably an easier sell as well if you come back to the concept of getting it across the minds of consumers and other stakeholders. The less assumptions you build in, I think the easier that gets. And so it'll be great to see that the once it's more fully fleshed out, to see what it looks like.
Yeah.
Okay, well, maybe we'll move to Arlen and whether you've got any questions maybe around feasibility or, as this goes to grant review panels, what your thoughts are.
Thanks, Steve. And thanks, Aidan. I thought that was a really, really fantastic presentation. Maybe I'll just start with some highlights. I really appreciated that. It seems like you're working up a strong collaboration with REMAP-CAP, and that federated platform idea is great. Appreciating the space with everyone gets a trial. It's nice to see us making steps towards this learning health system type approach. So I think that came across nicely. And I also thought that your pathway with that intervention selection criteria committee looked really good, to have some independence there, and especially appreciating seeing that being planned very early on. We've seen in platforms before that that's kind of a side thought that gets done after the first set of interventions. So it's good to see you thinking about that early. I want to make two main comments. One is on just on the back of Steve's comment earlier around that the device that you've got that you've mentioned there. If that device is not indicated to be able to diagnose what you need it to do or test that in Australia, just flagging the additional regulatory hurdles that you'll need to register that with the CTN with this trial. Even though it's not an intervention, it will still be kind of classified like one for the regulatory perspective. It won't add too much hurdles for you, but it's a bit of extra work in the early stages. And then you'll just need to think about how that might change as your platform goes on and that device then does get regulatory indication within that setting. Just you need to plan for those changes kind of early on. So have a think about that. I wanted to ask you about one of your exclusion criteria is prior enrollment within the trial, and the potential for patients to circle back through. You mentioned these two first interventions will be recruiting for probably 2 years, and then you've got some further ones planned. What percentage of patients are going to get ARDS again, come back in 2 years later, be readmitted, and they're a potential patient for you for the next intervention at that time? I'm just nervous, a bit of a shot in the foot if we're excluding everyone that might have previously participated.
So it's, you know, patients do get discharged from ICU and they do return. I think having, I think the percentage would be small. I don't have an exact figure, but you know, ARDS is a very severe critical illness, and you know, I would have to check the exact number, but I would say it would be less than 5% would be expected to come back with the same syndrome. It's usually, you know, the commonest inciting event would be like a infectious disease, a pneumonia, something like this, and you know, it's a little bit different than, say, chronic diseases like heart failure, where you do expect lots of patients to come back in again and again and again. So I don't think it will have a big impact on total numbers, maybe 5% or less, probably less.
Yeah, okay. That's good to know. If it's having a little impact then on your total numbers, but then there's the potential for just things to think about when training sites is, if someone comes back 2 years later and they're part of that 5%, how are we identifying that they previously participated in this trial and not re-enrolling them if you're going to keep that as an exclusion criteria? So just things to think about in your site training and how you're going to set up the longevity of the platform, really. Then just something that I didn't have come through strongly in your presentation that you might want to think about for your grant applications is I saw that you had your first interim analysis numbers on there, and then you had estimated timing. I think you said 6 monthly thereafter for your further subsequent analyses. What were your numbers in that in per period for that interim?
So it's obviously going to change over time as we recruit more sites. The goal is, I guess, once we're up to the full 70 sites, which is the goal, we think that we'll be able to recruit 2,000 patients over about 2 years. But we have up to 4 years to, you know, is what we're applying for. So that's the kind of recruitment rate that we're expecting.
So sorry, maybe I didn't articulate very well. My question was kind of, what is your N for each interim analysis post the first one? So your first one, I think you mentioned, is 240 or 80 in each arm. Subsequent to that, are you interiming every 30? Are you interiming every 60? What does that look like?
Look, I don't know the exact number that's been
Okay.
decided. I would guess it's going to be more than 30 or 80.
Yeah.
It's going to be more in the realm of, I would guess, a couple of hundred. But that's just a bit of a stab in the dark.
Okay. Yeah, all good. Just something to think about
Mhm.
to be coming through more clearly.
Yep.
But otherwise, I agree with Rob. I thought it was a fairly easy platform. Nice that it's open label. That makes life a lot easier for your setup and your sites, and particularly these first two interventions being fairly readily available medications. It seems like you should be able to get off to a really strong start, which helps platforms a lot.
Great.
Thanks.
I might hand over to Hannah. I guess, Hannah, particularly for phase 2 trials, how does the health economics bit fit into this? And do you have questions for Aidan? I might have missed it, but I didn't see a lot, Aidan, at this stage about, you know, a health economic analysis.
Yes. Yeah, so I didn't see anything about cost effectiveness either. So my first question, I guess, was going to be wondering if you've just thought about that, Aidan, but it isn't typical, I guess, to build in cost effectiveness analyses in phase 2 studies. Normally we would see them more just in phase 3. But it's not a bad thing to think about just in terms of looking at like if you had any concerns about feasibility of data collection in terms of the costs or the particular quality of life instruments that we like to use in cost effectiveness. Phase 2 studies are a good chance to sort of test the feasibility of the data collection if you did have any concerns about that. So that would just be, I guess, something to think about. But From what I can tell, the treatments in this particular study, I think, are quite cheap. Correct me if I'm wrong. So, and it seems like in terms of the outcomes, if treatments are more effective, they're likely to be cost saving in this case, because they'd probably lead to reduced ICU days or reduced hospital days and better longer term outcomes. So I think the case for cost effectiveness is probably quite strong if you were to look at it, but yeah, it's definitely not necessary. I think for a grant application in a phase 2 study, I don't think it would be an expectation, but might just be helpful to show that you've been thinking about it. So I don't know if that helps. And I did just have a question about what particular quality of life instrument you were using, Aidan. I think you said quality of life collected at 90 days. And I was just wondering what instrument you had in mind.
I know in ICU studies we often use the E5QD.
Yeah, EQ5D. Yeah.
EQ5D. Sorry, yes,
Yeah,
that's right.
perfect. Yeah.
For ICU studies, or we use the HUDAS. I'm not 100% sure which one's been decided, but it would be, I would guess, one of those two.
Yeah, sure. And I would just suggest, if you're using particularly the EQ5D, is one that we would recommend for cost effectiveness analysis, because that can be quite readily converted to quality adjusted life years. But for that to happen, it does need to be collected at a baseline time point as well as a follow-up. So something else to think about if you did want to look at in your analysis a quality adjusted life year estimate, if you built that questionnaire in at a baseline and the 90 days, that would be the way to go.
Brilliant. Yep. No, thank you. We do have some experience doing that, but I think you're right. The goal would be to incorporate all of this into the phase 3 trials once we start developing these new interventions. You know, understanding these broader issues are paramount. So, yep. Thank you.
Actually come to the end of our hour. I know we started a couple of minutes late. Are there any burning points that any of the other participants would like to make before we finish up? I think there's opportunities. You know, feel free to email Aidan directly if there's some feedback you'd like to give.
There is just a question in the Q&A from Tash, if you can see that, Steve.
I can't actually. Do you mind maybe if you could read it out, Vivian?
Yeah, absolutely. So Tash wrote, out of interest and from a feasibility of recruitment perspective, what is the prevalence of ARDS? How many are newly diagnosed per year? And is it different in Australia to the UK or other parts of the world?
It's a great question. I think I don't actually have the figure off the top of my head. I know that it would be in the realm of, you know, if I guessed, it would be in the realm of 5,000 to 10,000 patients a year. That could be wrong. If anyone else is online, please correct me. But it's a very common. So mechanical ventilation for acute hypoxic respiratory failure is very common in ICUs, and this is a common disease within that group. Like I said at the beginning, 20 to 25% of patients meet the definition of ARDS. So we're talking, you know, thousands of patients every year. There is a bit of a trick that it can be hard to diagnose, and there's a lot of variation between different methods. That could be a bit of a challenge for this. We will use the new definition, but there is some potential difficulties there. On the second part of the question, is it different in Australia to the UK? It does seem like there is less ARDS in Australia than in some other parts of the world. We have a very large global study that looked at the incidence, and it seems very high in other countries compared to what we see. So I think that's potentially a bit of a barrier for us in Australia, but I think overall it will be good for the trial because we have these high prevalence in other countries. It may be related to some of the way the hospital system is set up here, the larger ICUs, the lack of smaller regional hospitals. I don't know. Intensivists love talking about why this occurs, but yeah, there probably is some individual variation, but we're not 100% sure why.
Great. Well, thanks very much for a fantastic presentation, Aidan. very much good luck, and hopefully it'll be a successful grant application that comes up. It was really good to hear about what looks like a very well designed study at this stage. And thanks to Hannah and Arlen and Rob and to Mitch for all their inputs as well. Okay, we might finish up there. Thanks very much, everyone.